Welcome. My name is Colleen Williams, and I'm the Manager of Public Relations here at the Endocrine Society. We hope you're enjoying your experience at ENDO so far. During today's news conference, we'll be exploring the latest science on endocrine disrupting chemicals. We're pleased to have with us Dr. Christopher Casades of Duke University's Nicholas School of the Environment, Dr. Deborah Koresh of the University of Calgary in Canada, and Dr. Elaine Costa of Sao Paulo University in Sao Paulo, Brazil. Over the next 35 minutes, each speaker will present their findings, and then we'll have a brief Q&A session at the very end. Please note that the news conference is being broadcast live via webcast, and we do have several journalists that are watching remotely. So it's very important that when you do ask a question or make remarks that you please speak into the microphone. With that, I would now like to invite our first presenter, Dr. Casades, to the podium.
Hi all, thanks for being here. So today I'm going to share some of the work that we've been doing on the ability of indoor contaminants to promote fat cell development. So I'm sure you're all very familiar with the worsening obesity epidemic, both in the US and globally. So in the US, currently about 40% of adults are classified as obese. That is a lower percentage for younger individuals, so about 1 in 10 infants and toddlers classified as an obese, and about 1 in 5 2 to 19-year-olds. This contributes to more than $265 billion in U.S. healthcare costs each year, and on a statewide basis, that's about 10% of total state healthcare costs on obesity-related illnesses. And so one of the reasons this has really gotten a continued attention is that really interventions have produced only modest effects. And so there was this great paper in 2002 by Bailey Hamilton really demonstrating quite well that caloric intake, activity, and genetics were insufficient to explain the magnitude and the speed of this worsening health trend. And the other aspect of this paper really highlighted that there are actually a lot of environmental contaminants that have been demonstrated to modulate weight in animals and humans. So that's where our interest came in. So one of the things that our lab does is collect and measure contaminants in indoor house dust. So in this case, what we're doing is taking house dust samples from people's homes. We solvent extract that in the lab, and then we apply that extract to our kind of common pre-adipocyte cell assay, the 3T3L1 cells. And then we can measure two things from that. We can measure the ability of these contaminants or the mixture of contaminants present in house dust to drive proliferation of these fat precursor cells, or differentiation of these cells into mature fat cells. And we do that by measuring the lipid content of the cells. So we just published this work in Science of the Total Environment, looking at a cohort of about 150 people in central North Carolina. And we found that the extent of dust-induced fat cell development, so the extent that each dust extract drove the development of fat cells in our cell model, was correlated with the health of the people living in these homes. So the thyroid stimulating hormone in the adult residents was positively correlated with the degree of fat cell development, and free T3 and T4 were negatively correlated with the extent of fat cell development. And further, perhaps more interestingly, the greater the extent that the dust was able to promote fat cell development, the greater the body mass index of the people living in those homes. So definitely demonstrating a relationship between the chemicals present in the indoor environment and your metabolic health. And so the work that I'm presenting here at the meeting is following up on this in a child health cohort study. So this is the TESI cohort, about 203 children from 190 central North Carolina homes, collected from 2014 to 2016. Each participant had a home visit, and dust samples were collected as a measure of indoor exposure. This was then, as I mentioned before, solvent extracted and concentrated for application in the bioassay of fat cell development. And really our goal here was to evaluate the putative role of environmental contaminants present in dust on the metabolic health of the children living in those homes, and figuring out what are the causative chemicals driving those effects and what are the mechanisms that might be happening through. So to give you an overview of what we found that about 80% of the dust extracts promoted significant fat cell development. Some of those dust samples did that via differentiation of the cells into mature fat cells, which is what we measure via lipid accumulation. And others did that via proliferation of those adipocyte precursor cells. And then some do both. So there's differing cell populations. And we tested everything at kind of a set 100 microgram level. And to put that in perspective for you, children are estimated to consume about between 60 and 100 milligrams per day, so more than 1,000 times higher. All right, so looking at mechanism, we looked at two mechanisms that are known to affect fat cell development. So PPAR gamma is considered the master regulator of fat cell development. And when we tested our dust extracts for activation of PPAR gamma and correlated that with the extent of fat cell development, we didn't see any correlation, any significant correlation, suggesting maybe PPAR gamma might not be our causative factor here. When we looked at the thyroid receptor antagonism or inhibition instead, we found a strong positive correlation between the extent of thyroid receptor inhibition and the extent of fat cell development, suggesting that might be a contributory role. And I'll just highlight that that's the contributory factor that we found in our previous study that we just published, and we confirmed that in that previous cohort of adults via some silencing and other experiments. So we next looked at trying to figure out what are the chemicals driving these effects. And so I am in an environmental chemistry lab. This is one of the strengths of what we do here. So I have data on 111 different chemicals that were measured in these dust samples and found that 68 of those were positively correlated with the extent of fat cell development via the lipid accumulation of the cells. And then 13 of those 111 were positively correlated were positively correlated, 4 of them negatively correlated with the proliferation of the cells. And this graphic is just showing you that when we start looking at mixtures of these contaminants, most of these contaminants are having low-level effects. So really what we think is going on here is lots of contaminants with a small amount of effect on fat cell development, resulting in the effects that we're measuring. And with that, I'll stop and just thank everyone in our lab who's been helping on this project. So Kate Hoffman doing all the epidemiology work in our lab, Allison Phillips who did all the extractions of the dust, and then the Ferguson lab with Lee Ferguson is now assisting with the non-target analysis, so working out if we can measure 111 chemicals in the dust, what else is there that might be contributing to the effects that we're measuring. And I have a poster tomorrow as well where I discuss more of the non-target data and figuring out what might be significantly and enriched in the homes of people who are categorized as obese or whose dust demonstrates the greatest effects in the bioassay. So I'm happy to talk to you more about that later. Thanks.
Thank you very much, Dr. Casadas. I would now like to invite Dr. Koresh to the podium. Where are these going to be? Where's the...
Okay, well, thank you for inviting me to be here today. So I'm looking forward to telling you about some work that we've started to do looking at bisphenol A's effect on circadian activity using mice as our model system. So in the lab, we became interested in BPA several years ago. These are work were done, published around 2012, looking at the correlation between maternal BPA levels and the childhood behaviors later in life. So these were kids that were exposed to BPA while in the womb during gestation, and they found that moms that had high levels of BPA during their pregnancy, their children went on to have a variety of childhood disorders, most notably hyperactivity. And we became interested in, well, how does that happen? How does the developing brain know that BPA is present, and how does it change the developing brain? And can we measure it in the laboratory setting? And so BPA probably needs no introduction, but just to be clear, it's a plasticizer. It's used in a variety of consumer products. It's found in food containers. It's the lining of cans. It's found on thermal receipts, dental sealant, medical tubing, and a wide variety of plastics. So all of us, nearly all of us, have BPA in our urine at any given time, and it's nearly ubiquitous in our society. It functions through a variety of pathways. There's no mouse. Okay. As shown. Oh, but we're on. No, it's okay. Through a variety of pathways as shown here, and it's what we consider a messy compound. It actually functions in various ways depending on the local environment and the receptors that are present. So in my lab, we're interested in the hypothalamus. So the hypothalamus is a region of the brain. It sits at the base of the brain, and it organizes its neurons into these clusters. And so the hypothalamus is critical for your survival, and it is important for maintaining what we call homeostatic processes, which is basically sensing and stabilizing the body. So it senses, are you hot, are you cold, and adjusts accordingly. Are you thirsty, and adjusts accordingly. Are you hungry, are you full, so on and so forth. It also has roles in parenting and various attachment behaviors, in addition to the hormones that control the body function. In particular, the hypothalamus is important for circadian rhythms, and so it's important for maintaining your diurnal cycles. And so in our lab, we have shown that bisphenol A affects the development of the hypothalamus. We've shown it in fish, we've shown it in mice, and we can show that the neurons at the timing of when they are born seem to be particularly susceptible to bisphenol A, even more so than other brain regions like the cortex or the hippocampus. And this perhaps isn't surprising because the hypothalamus is known to be sensitive to testosterone and estrogen, and bisphenol A acts in a similar manner. And so it's perhaps is logical that it would have an effect particularly in this brain region. So we became interested in the role in the effects of bisphenol A on the circadian rhythms and sleep as it is set up by the hypothalamus. So the region of the hypothalamus that controls your circadian rhythms is called the suprachiasmatic nucleus. We'll refer to it as the SCN. So the SCN is entrained by light, and so what that means is that light will come into your eye, it will travel down your retinal hypothalamic tract, and it will put inputs onto your SCN, which then is responsible for controlling your diurnal cycle. So your sleep-wake cycles as well as other processes throughout your body that is responsive to this type of circadian activity. So like most nuclei in the hypothalamus, it has a complex morphology and structure. And so there's a core region where certain neurons reside, and then there's a shell region where different neurons reside. And these two populations work together to control your circadian cycle. It's a very intricate process where proteins work together to give you this type of cycling that then is sensed throughout your entire body via projections that are spread to other regions of the brain. So the way we study circadian activity in a mouse model is by measuring the activity of the animal. And so what you see in the upper corner here is a running wheel. And so we can put a running wheel inside the cage, and unlike you and I and our beloved relationship with our treadmills, the mice really like these running wheels. And so they get on them, it's an enrichment tool, they think it's great, they're very, very happy. They will just run, run, run. And so what we can do is we can measure how much activity they are having by every time the wheel goes around a circle, it's going to give a click, and we can quantify that. So what you're looking at here is the gray bars are 6 hours of dark, and then you have the white is 12 hours of light, and then you have 6 hours of dark on the other side. And mice are nocturnal animals, as you can tell by looking at this. So each time the wheel goes around, we get a black slash. And so each row is a day. So this is like, I don't know, a couple weeks of data here. And so you can see, if you start at the light, they don't do much activity during the light cycle in the white spot because they're sleeping and they couldn't be bothered. But then as soon as the night comes, they're nocturnal, they wake up and the first thing they do is they get on their wheel. So you can see as soon as dark starts, they start running. They run, run, run. Now, if you flip around to the latter part of the night, they start to get tired and they slow down. And eventually they think, okay, well, light's going to come soon and I'm going to go back to sleep. And then it starts over again. So we can quantify not only their sleep-wake patterns, but their activity by tracking how much they're running in their wheel. So when we take a mouse that was exposed to bisphenol A during gestation, we see something different. So just to be clear, we exposed a pregnant mouse to bisphenol A, and so what we're measuring is her offspring later in life. So these offspring had BPA when their brains were developed, and then it was washed out, they were grown, now they're adults, we put them in this assay, and they haven't seen BPA for several months now. And what you can see is that there's actually more activity. So here, they have similar activity maybe to the wild type, but that it continues further into the night. So they stay hyperactive and in their wheels longer. And so we can quantify this, and you can see that there's an increase in their total activity, and there's an increase in their duration of activity. So when they are active, it's for longer periods of time. So this suggests somehow exposure to BPA during gestation has some sort of lasting effects, that they are more active during their nocturnal cycle. So the question is, well, how does this affect their circadian activity? And to do this, we put the mice in total darkness. So here they're exposed to light, and then you take away light, and then you look at their activity. So as I said before, our eyes and the light entrain us to a certain cycle, but we have our own intrinsic cycle. So blind people are still able to have a circadian rhythm. It follows their own natural intrinsic cycle, and it's independent of what's happening with daylight savings or any other artificial influence we put on ourselves from the light cycles. So you can see that it starts to shift a little bit as these animals develop their own natural intrinsic cycle, but it's usually pretty consistent. So instead of it being a 24-hour cycle, maybe it's 23.5 or 23.6 or so on. What you can tell in the BPA-exposed animals is that the slope is considerably different. You can tell that this looks different than this, suggesting that their circadian activity has been altered. And so when they are exposed to dark only, you can see there's an increase in total activity, which is what we just showed when they had a light-dark cycle, and that their duration of activity is increased, which is also consistent with what we just showed. But what's remarkable is that there's a change in their circadian period. And so that's what's measured in the slope here, that their days are actually shorter than what you would have for a control animal. And so they shrink down their circadian periodicity. We became interested in why this might be happening, and so for this we turned to vasopressin and other neurosecretory cells that are in the suprachiasmatic nucleus. And what you can see in the wild type, that there's these vasopressin neurons that are largely absent from the core. They should be present here in the shell. And in our BPA exposed, you can see that there's an increase in vasopressin staining. And this is perhaps not surprising because vasopressin plays two roles. Well, it plays many roles, but the two primary in relation to this project is that it sends outputs to other parts of the brain region to control cycling of the circadian cycles elsewhere, but it also stimulates the neurons in the suprachiasmatic nucleus to get the clock machinery going of the circadian core. So having more vasopressin neurons could somehow be altering how the suprachiasmatic nucleus is controlling circadian cycling in these animals. And so that leads us to our general working model, which is if you have a pregnant mouse and you expose her to bisphenol A, that the BPA will cross the placenta, it will then enter into the developing brain, it will alter the developmental programs of that brain, such that in this case the suprachiasmatic nucleus has different quantity of neurons, so it has more vasopressin neurons, and they also seem to be mislocalized to the wrong regions, and that's going to affect, if you have more neurons, you're going to have more projections projections. That's what neurons do. They send projections, and it's going to affect how it stimulates other brain regions, and it could lead to hyperactivity, which is what we're measuring in their running wheels. So with that, I want to thank the group. This was done by Dinu Dasan, a postdoc in the lab who spearheaded all of this work in collaboration with Mike Antle, also at the University of Calgary, who's been a big assist in this project, and then our funding. So thank you.
Thank you, Dr. Koresh. I would now like to invite Dr. Costa to the podium.
Good morning. I'm pleased and honored to be here today, and I'd like to thank you for inviting me to present our research that is exposure to fine inhalable particle matter of air pollution. PM 2.5 is a teeny particle of the city of São Paulo, causes tubular degeneration and altered mitochondrial-related gene expression in mice tests. This work was performed in collaboration of 3 labs in the University of São Paulo, and Catarina is my fellow who developed all this work. So, air pollution is defined as the presence in the air of one or more substance at a concentration or for a duration above their natural levels, with their potential to produce an adverse effect. Anthropogenic activities have led to increase in many air pollutants to levels that have adverse impacts on human and environment health. In urban areas, vehicular and emissions in industries and power plants are the major source of thousands harmful compounds emitted into the air in the complex mixture composed by gas, polycyclic aromatic hydrocarbons, solid particles that we named here particulate matter, and especially particulate matter 2.5 with 2.5 micrometers. And have metals. PM 2.5 micrometers is a fine inhalable particles with 2.5 micrometers or smaller. All of these compounds are known to disrupt in the endocrine system, including male reproductive system in human and animals. So they are called endocrine disrupting chemicals. What definition of endocrine disruptive chemicals is exogenous substance of mixture that alters functions of the endocrine system and consequently cause adverse health effects in intact organs or its progeny or subpopulation. Moving on to fertility. We know that in modern industry societies, human male infertility is declining, whereas the incidence of disease or birth defects of male reproductive systems is increasing, such as hypospadia and cryptorchidism. Hypospadia is a normally placed urethral opening at the underside of the penis, and the cryptorchidism is undescendant tests in men. Also, the World Health Organization estimates that approximately 15% of the global population has difficulty with fertility, and male infertility accounts about half of those problems. Based on this data, our aim was to evaluate under different conditions the effect of exposure to air pollution of the city of São Paulo on mice tests. Our study included 4 groups of animals. The animals born from clean progenitors. The first group, named control group, was exposed to filtered air during gestation and post-weaning time until adulthood. The second group was exposed in prenatal, were exposed of PM 2.5 human particles from São Paulo City only during gestation. The third group was exposed before and after birth, from during the gestation and post-weaning time until adulthood. And the fourth group, post-weaning group, was exposed only during after weaning time until adulthood. Here you can see our chamber of exposure. It was built on the parking place of the Medical School of the University of São Paulo, and it was developed by School of Public Health of Harvard University. In this photo, we have in this photo, we can see a kind of accelerator that capture the pollutant of the air and distribute to here, and distribute to exposure chamber here and back. Here we can see the animals in the cage during the exposure to an amount that was similar that an adult breathed in 24 hours in São Paulo. Our results showed us that the tests were heavier, especially in prenatal post-weaning group. The test was heavy. They are bigger, especially because the accumulation of fat in these testicles, but also the epididymis weight was higher in this group, and germinal epithelium volume was higher also in this group in comparison with other group. Here we can see in the histological analysis, we can see that the tubes in the tests that produce sperm of all the exposed mice showed signs of deterioration, especially in prenatal post-weaning group. Here we can see the disorganization of spermatogenesis, and the sperm of prenatal post-weaning group has worse quality. So here in this figure is showing you seminiferous tubulus from testicles for post-weaning group. The worst one was prenatal post-weaning group, but here I show you the post-weaning group. And we can see in the black arrow the abnormal residual bodies, these bigger cells, and spermatid retention. Here we can see the spermatid placed here, and instead here in the lumen. We can see in red circle the multinucleated giant cells, is accumulation of cells, and germ cells disorganization, okay? But beside that, the elongated spermatid we can see here in the green arrow that we have elongated spermatid ready to be spermiate. So we have a few amounts of spermatid ready to spermiate. So all these alterations demonstrated that air pollution can disarrange the spermatogenesis of male mice, particularly in animals exposed in pre and postnatal time. Moving on to the gene expression, we found that genes related to mitochondrial RNA related exhibit differential expression between the groups, especially in the exposed group. Particularly this gene, MTTY gene, was found to hyperexpressed in prenatal post-weaning group in comparison with the other group, as you can see in green bars. So, in conclusion, we demonstrated that the first time that the exposure to PM 2.5 of the city of São Paulo caused tubular degeneration with decreased or absent spermatogenesis associated with differential expression of genes related to mitochondrial function in mice tests. In addition, we demonstrated that postnatal exposure seems to be more deleterious for the testicular functions. Thank you for your attention.
Thank you so much, Dr. Costa. I would now like to open up the mic for Q&A. If everyone could please state your name and affiliation before asking your question, and remember to speak into the microphone, that would be great.
Hi, Katie Calvitis with Endocrine today. Dr. Casadas, can you speak to some of the future research in this area with the chemicals and dust, please?
Sure. So one of the paths that we are following up on is, as I mentioned, the non-target analysis. So you can think of this as kind of two ways of looking at figuring out what chemicals are driving effects. You can measure the concentrations of specific chemicals that you are targeting, or you can look at the greater chemical space. So this is work that I'm doing with Lee Ferguson at Duke, and so our preliminary look at house dust finds about 35,000 different chemicals in house dust. So it's really, when I say, through the work in Heather Stapleton's lab, we're looking at, measurements of 111 different chemicals. That's impressive, but still a very small representation of all the other chemicals that are there. So that's one of the areas that we're going next, trying to figure out which of those other chemicals might be relevant to the effects on fat cell development that we're seeing. And then we're also definitely still looking at trying to better sort out what are the mechanisms driving that. We think that probably some of it's thyroid mediated. And then also looking at the health effects in the people living in those homes. Thanks.
Hi, Diana from BMC Medicine. My question's for Dr. Kurash, if I'm saying that right. So in terms of the findings that you presented, are they potentially reversible by targeting vasopressin or adjusting light cycles?
It's not clear, I think, is the best answer. It suggests that BPA is somehow influencing how the region develops, which means it might be hard to change it, but maybe not impossible to overcome. I don't know. We haven't gotten that far in the research to look at ways that you would treat it or dose it to reverse it.
Okay, I don't think we have any other questions. So thank you to all of our speakers and to all of you for coming out today, and we hope you enjoy the rest of your time at Endo. Thank you very much.